So in last lecture, we have seen basics of biosensor or introduction to biosensor till component of biosensor. The components of biosensors which we have seen in bit short. So this is what components of biosensor or a block diagram of a biosensor look like. It consists of analyte, bioreceptor, transducer, electronic part, and a display where we want to see the output. This is, we can say, that schematic or a block diagram, or you can say the component of a biosensor. Now its representation we have seen in a short, like what it individual component is used for. Now we'll see these components of biosensor in large, what each component is useful, what it consists of. So the first component which we have seen in the previous slide is basically Analyte. Now we can see from the biosensor diagram, component diagram, the first and foremost is Analyte. And this Analyte basically is the part which is most important to us. Let's say if you want to measure uh, blood glucose, if we are interested in monitoring blood glucose. So from the blood glucose, the glucose is Analyte. That is what we are interested in. So Analyte is the first part from our component. The next part is basically bioreceptor. So these are a few bioreceptors as we see in the component. So what is bioreceptor basically? So the Analyte is basically interacting with something. So this target Analyte interact with biological component, and that biological component is called as bioreceptor. Now this bioreceptor is the second component which is of a biosensor. So this bioreceptor is specifically designed in a way that it will communicate or interact with the particular analyte of our interest. So when this analyte interacts with or binds with specific analyte, it will produce a measurable effect at the output of bioreceptor. And that measurable output, we can take it as a for the input as a next step for the transducer basically. So what is the need, or we can say what is the peculiar characteristics that drives the bioreceptor? Now that is, take an example, my blood may consist of different kinds of sugar. It may consist of glucose, it may consist of fructose, it may any other uh, sugar. But I'm mostly interested in finding out the glucose content. So if a mixture is given, any homo or heterogeneous mixture, my biosensor, my bioreceptor has to pick a particular analyte and bind with it among a different combination of solutions. So for bioreceptor, selectivity has to be very high. So high selectivity is the one of the most important characteristics possessed by bioreceptor. So these bioreceptors can be classified according to the what kinds of common types they are used, classified accordingly, like how what kind of interactions they are involving, whether it is antigen antibody, whether they are enzyme ligands, whether nucleic acid, DNA, cellular structure, cells, or any biomimetic materials. So we can see this combination here, say antigen antibody, ligand receptor, DNA cell, enzyme substrate. So these are few types of bioreceptor present in bio sensors. So here the analyte is interacting or gets uh, communicated and interacted with the analyte, and reaction happens. So what is the most important function of bioreceptor? So as we have seen, the most important function of bioreceptor is to interact with a specific target compound. So that is the target compound analyte. That is what we are mostly interested. So the compound which is to be detected is the most important to us, and that has to be taken care. So this is most important function of a bioreceptor. So secondly, high selectivity. So the bioreceptor has to be able to detect the presence of my target compound in a particular given solution. So already we have seen the example. Next, it should have ability for that biological element to interact specifically with the target compound. So it should have a specificity also. So this specificity forms the basis of my biosensor. So it is the one of the most important characteristics. So what are the different types of biosensors? So already we have just seen few. So these are types of bioreceptors: antigen antibody interactions, artificial binding proteins, enzymatic interactions, affinity binding receptors, nucleic acid interactions, epigenetics, cells, tissue level. So these are different types of bioreceptors which might be used. So the next component which present in biosensor, components of biosensor, is basically a transducer. So I have now received the combination of chemical reaction which has happened between bioreceptor and analyte, specific analyte. So the interaction is happened, some chemical reaction happened, and I got some signal, physical signal. Now next part comes, that is most important. Now the transducer. So basically this transducer is a basic part device of or component of a biosensor which converts whatever biological signal produced by bioreceptor, and we can say that the interaction between analyte and bioreceptor produces some signal, and that signal now needs to be converted into some measurable signal, and this transducer does that basically. So it acts as an interface between bioreceptor and the next part, that is maybe any electronics part which is we are using for further processing. So it basically serves as interface between basically interface which measures the physical changes occurring because of at bioreceptor end, and converts those energy what are created into a measurable electrical signal, so that we will be able to process that electrical signal in a better way, and we can have processing of that electrical signal. So this signal can be in the form of electrical, thermal, mechanical, it may be optical, or depending on what kind of sensor we are designing, what application we are processing, a lot of things will get involved as we go into deeper what kind of form of that particular signal will be. So now this transducer plays an important role. So what kind of transconduction mechanism which we are using in this transducer? So it may be pH change happening, it may be heat generated, it may be light proton, it may be mass change happening, it may be an electroactive substance present, it may be a pH change. So depending on what kind of transconduction mechanism happening, the transducers we are having different way to using. So if I am having a pH change of my solution, I may go with a semiconductor based pH electrodes. If there is an electroactive substance, I may go with a basic electrode. If there is a change in mass happening before the reaction and after the reaction between the components, I may use an piezoelectric device which works on the piezoelectric effect. If there is heat generated, then I may use a thermistor. If there is I may use a photon counter detection for light. So depending on what kind of transconduction mechanism is my system is using, I can choose transducer's typical component or the type of transducer I may change. So it depends upon a particular application, what kind of transconduction mechanism is I want to use. And this transconduction mechanism is basically what output I am getting before transducer. That's what kind of mechanism I have to use. If there is a heat getting at the output of a Bioreceptor, I may go for a typical type of sensor here in a transducer. So this is the same slide which we have already discussed. Here it is presented in a different way. What kind of transconduction mechanism, what are its types, and what happens in basically in those spatial categories. So the transducer is present. I have an optical transducer. So what dependent mechanisms? It may be an absorption phenomenon used, fluorescence may be used, interference of light is used. So what happens here? We basically measure the changes which is happening in a light intensities. So whatever light intensity changes, we measure those changes and appropriate signal is given to the next processing. So here may have electrochemical signal. So we have potentiometric, amperometric, conductometric, depending on whether voltage current is developed at the electrodes. So the whatever electric distribution is used as a measurement tool here. Now we have mass based, that is piezoelectric detection based system, a transducer. Basically, we have some in next here we use cantilever beam, micro cantilever beam. There we use a piezoelectric changes. So whatever mass change happen because of the pressure, we use this kind of technology. So change in the mass of analyte, we measure those changes and we'll use this such type of a transducer in some mechanisms. We have a temperature based like calorimetric detection devices. So whatever temperature changes, we are correlating with the signal. And last, which or one of the next is electrical and magnetic properties we may use. So there may be dielectric permeability properties we have to check. So in short, we will look for electrical and magnetic properties of a typical analyte and we will process those typical values. So these are the transducer types which we have already just discussed. Electrochemical transducers, which basically measure the electrical changes in the electrical properties. Those electrical properties may be current, voltage, impedance. So used in biomedical application, glucose monitoring. We have optical transducers, so which measures changes in light. We have already seen reflectance, absorptions, fluorescence. So used in optical transducers. We have a piezoelectric transducers. So basically this special category measures the changes in mass or pressure by detecting vibrations or resonance frequency changes. So this is basically used in mostly in sensors detecting DNA or other new molecular binding events. So we have next is thermal transducers. So whatever heat generated to measure changes in heat because of biochemical reactions, typically used in calorimetric biosensors. And then we have magnetic transducers. We use the magnetic properties, often basically used in applications like immunosensors, where we have magnetic bead structure. So those are the different transducer type in short, which we have already seen now. Now, the next basic component of a biosensor is a signal processor. Now, whatever signal I have received, electrical signal after the transducer, I may need to process it. Now, consider an example which we are taking an heart beats. Now, the sound of heart which I have received from the sensor, the values will be very big. There may be a noise present. So, I may need to filter it out the noise. I may have to pick only the particular heartbeat signal and I have to process it. So the signal processing part comes into picture. So the signal processor unit gets the signal from transducer, make a process that particular signal, convert those signal into a particular readable unit, interpretable output, and gives us to human readable format. Now, these steps may involve different mechanisms. Now, my signal may be weak, so I may have to amplify the signal. So amplification comes. So amplification basically strengthens signal for easier detection at a later stages. I may use a signal conditioning. If there is a noise present, I may filter it out, modify the signal to eliminate the noise, and I have to increase the improve the accuracy. So in such cases, I may have to increase my signal to noise ratio. So noise will be suppressed and my signal will be amplified. Data processing, I may have to analyze and convert the signal into some usable form, maybe in numerical data, graphical display, so that I can show the data to real world, how the data processing happens. And in particular, some cases we may have to integration with a computer or a digital display to provide a real-time information and analysis. There are different ways, like when it is used in uh, ICUs, we may see the typical real-time monitoring. So those are the cases. So the signal processing is the most important part here also, because we have to process the signal, whatever we have received in a processing, and then we have to get the output in an appropriate format. Now the next important device at the towards the end of the biosensor component, we get a display or output device. Now it depends upon whether we want a quantification or only detection purpose, what purpose I am using my biosensor. So depending on that, my display devices may vary. So what is basically display or output device? In majority of the cases, whatever output I am getting from uh, transducer signal processing end, I need to present it to somebody so that that particular person user may check the output and understand it for whatever action you want to take according to this. So this display or output device could be visual displays, audio signals, data storage unit I may have to use for further processing. So visual displays may be like LED, 7 segment, digital readouts, graphs, so point of care devices we use most of the time. So those are visual displays. I may use some cases audio signals. I may have to create some alarm, buzzers. So some sensor provide an audible output tone depending on a particular conditions met by that output devices. In majority of the systems, these audio video signals are used. But in some complex system, I may have to refer the back data for further analyzing process. In those cases, I may have to store this data for later analysis, or may have to use cloud, and I have to share the data to the cloud, save it on a mobile or cloud-based servers. So best example is we use step counters. So the data is stored on a mobile phone or cloud-based servers. So this is display or output device. So these are what major components of a biosensor. But few components which we use different mechanisms. So those comes into categories. These are typical categories or applications of biosensor. So they need supporting components. So the few supporting components mentioned here, sample delivery systems. So in few biosensors, we need to some kind of mechanism where we want to deliver the sample from one place to another place. In blood glucose, air, water from one. In blood glucose monitoring, we need a strip so that whatever blood we got in, and we have to transmit it to the other sensing area. And this can be a microfluidic chip or a manual system. Second is basically a power source. Now this is what most important part in supporting component. If your standstill device which put in one place and you are doing some analysis, you may use a AC source of signal power. But in the majority of the devices now we are using handheld devices, so we have to operate on a power which is DC, and we don't have many or times charging. We have to reduce those. So in those cases, we may have to provide a variable or portable cases. We have to provide a DC power source. So in those cases, we have to use a batteries or external power. So depending on that, my biosensor again is classified in those ways also. Then we have membranes or filters. So like I want to remove some impurities, some unwanted substances. So I may have to use filters or membranes to remove those unwanted samples. So that only target analyte will be interacting with my bioreceptor, and no other any similar kind of component is interacting with my bioreceptor. This is the different supporting components which I have to. So why I have to use supporting component? Basically, because in some applications, so this is depending on application, case to case basis. So in different applications, I may have to use some different ways of mechanisms, like power, how to deliver the, what kind of filters I have to use. So in those cases, I use supporting components for those particular cases.